Hello scientists! Today we're going to talk about medicines that we use after an infection has already begun. Our big idea for this lesson is that medicines can work alongside the body's defenses to reduce symptoms or eliminate infections. Antibiotics and antivirals are two types of medicine that work to kill off the germ, whether that is a bacterial or a viral germ. So antibiotics specifically target bacterial cells, but not host cells. So they're going to kill off the germs, but hopefully, if it's a good antibiotic, not your germs, or not your cells. They target different parts of the bacterial structure, such as the cell wall being made, or different proteins being made, or certain chemical reactions that happen in order to keep the bacterial cell alive. So different classes of antibiotics work by targeting different parts of the bacterial cells. And it's important that there's something specific about that, whatever is happening in the bacterial cell that's not happening in your cells. So if it's a particular chemical reaction, one that's only happening in the bacterial cells or is facilitated by a particular enzyme, so that the antibiotic can tell the difference. So some examples are penicillin, amoxicillin, doxycycline, ciprofloxacin, and azithromycin. And one thing that we can note is that certain types of antibiotics have different endings to their names. So penicillin and amoxicillin both end with -cillin. They're in the same sort of family, and they act in similar ways. So antibiotics work on bacteria, and antivirals work on viruses. So antivirals work by preventing viruses from getting into cells and or from preventing them from multiplying and then taking over more cells. Some examples that you might have heard of are Tamiflu and Simitrel. And one important thing to keep in mind is that antibiotics don't work on viruses and vice versa. So if you have the flu, you are not going to, which is a virus, you're not going to take an antibiotic. And if you have an infected cut, you're not going to take a antiviral because you're probably have a bacterial infection. We can think about medicines as any molecule that we are introducing to our body that's going to bind and interact with antigens, right, or proteins. In this, for medicine, we often call them receptors on a cell, whether it's your own body cells or it's the cell of a pathogen. Some drug molecules bind to cells and send a signal to increase or decrease the production of other chemicals. These kinds of drugs are usually working on cells that are within our bodies, and often our immune cells. So you might take Benadryl if you needed an antihistamine, if you were having some kind of swelling or allergic reaction. And how Benadryl works is it binds to histamine receptors on mast cells, which are cells in your immune system. And normally mast cells, if they have a signal to produce histamine, they're going to make histamine, which then is going to go and increase blood flow and then cause inflammation, which we talked about previously. But when Benadryl is bound to those histamine receptors, it stops the signal to produce histamine, kind of gets in the way of that signal. And what happens is now histamine isn't produced, and so you're not going to, if you took Benadryl, you're not going to experience that increased blood flow and inflammation. So it's binding to a receptor or an antigen, and that means that the signal is blocked and the chemical is not produced, in this case histamine. We also we do have drugs that bind to a receptor and create a signal, right? They send a signal to create more of a certain type of chemical. And then some drug molecules bind to antigens on germs or pathogens to target them for destruction or to destroy them. And penicillin is a good example of this. So penicillin binds to antigens on the cell walls of bacteria. And when it's bound, it prevents the cell wall from forming properly, which ends up killing the bacterial cells. And similar to the lock and key analogy that we've used to talk about antigens and antibodies, drugs kind of have, or they do have, a very similar structure and then the way that they interact with the antigen. So we need a drug molecule that is a match, a key, to fit the lock. So drugs in the same family have similar shapes, which means that they'll bind to similar receptors, right? They'll fit in similar locks, and therefore they're working in similar ways. They're sending the same kind of signals. So when scientists are trying to make a drug, they're thinking about this lock and key analogy. And they need to find the right shape of lock, right, the right molecule to fit the receptor. So sometimes scientists know the shape of the receptor, the shape of the lock, and they build a molecule to fit inside of it. Sometimes they know the shape of another molecule that binds to that receptor, right, a key that also fits, and they can build a molecule that's a similar shape, something that will also fit. And we know that all of these things are really, really, really small, and so ultimately this takes a lot of trial and error. Scientists will try something, they'll see if it works, they'll adjust the shape a tiny bit, see if that works, and on and on and on, until they find a molecule that does what they want it to do. That's it for today. We're going to talk about more of these concepts in class, but if you have any questions, please jot them down or leave them in the comments, and we'll talk about them next week.